Basically, how does the cell division take place? It doesn't get like automatically tear apart, or someone pinch it in half, or we can tear it. No, it is done by the distribution of genetic material, which is known as the DNA, to progeny, or in this case, we can say two daughter cells. But in the special case, which is known as meiosis, it takes place differently with the help of eggs plus sperm, which we'll talk about later. So, how does DNA store information? For this, let's take an example. This is a cell, for example. This has DNA. It stores some information. We call it genetic information. It is known as genome. In the genome, the genetic information is carried away, and it also is transferred into the progeny. So, in general, what basically happens during the cell division, for example, the DNA. In the next step, the cell copies its DNA. Later on, two daughter cells get the same identical DNA from its parent. You can say half here, half here, half here, half here. It is not that usual that they can get the same amount of material, but in the case of prokaryotes, it is identical, which means nearly same. Now the term comes genome, genetic information of DNA. It is known as genome. When DNA and 2 DNA, 3 DNA, 4 DNA, 5 DNA come into a line or form together itself, it is known as strand. Now what happens? For example, this is a one strand of DNA. Okay, and later on it started to get condensed, and at the last stage it is finally more condensed and tightly packed into structures with the help of proteins and other materials, genetic materials. We call it chromosome. This material we call it chromatin, that's previous stage, and this is single strand of DNA. Here multiple strands attach, and at last this forms occur. How the chromatin is formed? The chromatin is formed when the proteins plus some DNA strands combine together. This is the final stage before cell divides. Now, in the eukaryotes, species has some special number of chromosomes, or we can say some characteristics of chromosomes found in the cell nucleus. For instance, here is an example of human somatic cells. Here, total 46 type of chromosomes are found, or 23 in pair. But here's exception. One pair or two chromosome is different than the rest. They're known as sex chromosome or gamete or reproductive cells. For easier view, for example, this is one DNA or one chromosome, this is another chromosome. These two are in pair. We call it one pair. Okay, so now 23 pairs are found in each somatic cell. This is one chromosome. I found that people are confused, like what is one pair, one chromosome. So this is that. So basically, what are gametes? See, gametes are sperms and eggs in eukaryotes. I have a better picture for this one is known as sperm. This is the egg. These both take part in fertilization, which helps into cell division. What is fertilization? For now, we can basically say it is type of cell division that starts after the gamete mix together and form a new cell. Distribution during cell division in eukaryotes. So what happens when the chromosome get distributed in the cell division? Suppose if a cell is not dividing, this is cell, it is not dividing further, but it is in phase to divide. Sometimes what happens here, if it is a DNA here and there is a DNA here also, what does the cell do? Instead of dividing, it doubles its DNA or replicated DNA for the next cell generation. For this means to happen, they form a thin long chromatin fiber. For example, this one we can say is chromatin. Like I have explained you earlier, they form, it is long, it is thin. Furthermore, what happened next? After replication took place, what happened here? Before application, we can say it BR before application, after application AR. What we can observe here, you can see the chromatins are thin, long, but after application of DNA, we can see the chromatin are thick, more coiled, and we can see it with light microscope, which means they are visible now. So what we can conclude that like chromatins are not visible. The visibility only comes at the or after the replication phase, or when we observe or see chromosomes, the visibility comes. Suppose this is one chromosome. Okay, it is this one part, it is second part. We named it as chromatid. This one also chromatid. These both constitute together and we call it sister chromatids. They are joint copies of original chromosome. 50% here, 50 here, we can say that. Now when the two chromatids join together, for example, this is one chromatid here, this is two chromatid here, or we can say, wait, this is one chromatid here, this is two chromatid here, they are joined together. Okay, and we can say this line of them, it is attached by DNA molecules which are identical or nearly same by making a bond, and it makes type of stick on. We can call it cohescence. And this attachment, like these two chromatids are attached together, this attachment is known as sister chromatid cohesion, which means the two chromatids are now fixed together. They are stick together. They cannot be separated right now. Now another point. Each sister chromatid has a thing called centromere. What is centromere? For now we can say it is a region like where the both chromatids are attached closely at this point. We can say it's centromere. When the replication took place, it resulted into two sister chromatids, and these are duplicate chromosome. 50% here, 50% here, duplicate. And this also shows that it is made up of repetitive phases. We can say repetitive or nucleotide for DNA joined together in a repetitive manner, which gave the path for creating a centromere. So we can finally conclude that the centromere is the main glue point between the sister chromatids. So now on, this attachment is made by proteins. This attachment is made by the proteins which recognize and bound to the centromeric DNA. For example, this is centromere. It has some DNA. Okay. So the attachment caused by this is due to some proteins, which we'll study in biomolecules, which help in binding the DNA. Also, other proteins helps to condense the DNA. For example, this was DNA earlier, and later on it get condensed. It is done by several proteins also. So due to this, what happens next? See, for example, this is one chromosome here. This is the point we can say it's centromere. Yeah. At this part, we can say the DNA is getting condensed. This part is getting thinner, narrow. This part we can call it as chromosome arm. It is an arm of chromosome. Now in the both chromosomes, for example, wait. in this chromosome, this arm and this arm, we can separately call them as arm of the chromatid. Arm of the chromatid. So what happened next is the division took place. I have a better picture for this. So what happened here, see, you can see here is a centromere, this point, it is a chromosome arm that I told you above, and the duplication, or we can say DNA replication and condensation start. What is replication? I told you earlier, doubling of DNA. Condensation, thick coiling. Okay, here the division, or we can say the duplication, doesn't happen yet, doesn't begin yet. Here it is duplicated. Okay, now we can see that 50% here, 50% here, sister chromatids. Now what happens here, the division took place into two chromosomes. Here we can call it two daughter cell right now, as this process is called as mitosis, the overall process top to bottom. So actually what happened here, see, sister chromatids here, these were joined. What happened, they separated into two, one here and one here. Okay, so these are now individual chromosomes. They are no longer as called as sister chromatid. Why? Because they each share 50% of their parent cell, and the chromosomes, it's doubled during cell division. The cell division took place here, individual chromosomes, the new nucleus here in these cells receive chromosomes which are identical. Identical means nearly same of the parent. Now what happened here, like the sister chromatid like converts into individual chromosomes, and like the cell division happened here. This process we can term it as mitosis, because like the genetic material got divided here, the nucleus, and also after that cytokinesis happens. What is cytokinesis? Let me show you. So in cytokinesis what happened is, suppose this is a cell, first stage, it is cell, it is nucleus, wait, it is nucleus, the cell just place, it is cytoplasm. The nucleus continues to divide here. See, in this one the cytoplasm started to divide and the nucleus is also dividing. The nucleus is doubling. For example, this is n, this is 2n, it is getting in half, n and n here. After that, when the cytoplasm divide, the two cells get separated, two new cells form. We can now term this or coin this term as cytokinesis, which means division of the cytoplasm.